Hey there, medical students. Welcome back to our channel, where we break down complex biochemical concepts into easy to understand bits. Today, we're diving into hepatic glycolysis, a crucial metabolic pathway that happens in your liver. Let's get started. This is the outline of what we will look at today. 1. Reaction steps of the glycolytic pathway. 2. Energy yields from the glycolytic pathway. 3. Alternative fates of pyruvate. 4. Anabolic pathways associated with hepatic glycolysis. 5. Regulation of glycolysis in the liver. This includes the hormonal regulation of glycolysis, the regulatory enzymes, and the key activators and inhibitors of hepatic glycolysis. 6. Disorders of the glycolytic pathway. Now, when glucose enters the cell, the first thing that happens is that it is phosphorylated to form glucose 6-phosphate, as indicated on the screen. This helps to trap glucose into the cell so that more can flow in from the blood plasma. The enzyme that phosphorylates glucose is known as hexokinase. In the liver and beta cells of the pancreas, a special isoform of hexokinase, known as glucokinase, is the one that does the phosphorylation. We shall talk more about this enzyme later. The reactions of glycolysis can be grouped into two phases: the energy investment phase and the energy generating phase. In the energy investment phase, 2 moles of ATP are consumed as indicated on the screen. After glucose is phosphorylated to form glucose 6-phosphate, glucose 6-phosphate is isomerized to fructose 6-phosphate by the enzyme phosphohexose isomerase. Then, PHOS-PHOFRUCTOKINASE1, or PFK1, uses another ATP to phosphorylate fructose 6-phosphate, thereby forming fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is subsequently cleaved into two products: glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzyme that catalyzes that reaction is known as fructose 1,6-bisphosphate aldolase, or aldolase A. Both glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are isomers, and they are interconvertible. To continue with glycolysis, the dihydroxyacetone phosphate generated is converted to glyceraldehyde 3-phosphate by the triose-phosphate isomerase enzyme. The energy generation phase is displayed on the screen. The enzymes that catalyze the reaction steps are indicated on the screen. Here, you can see that 1 mole of NADH and 2 moles of ATP are generated from 1 glyceraldehyde 3-phosphate. The process of generating ATP in chemical reactions such as this is known as substrate-level phosphorylation. However, to get ATP from the NADH generated, the NADH has to be oxidized in the electron transport chain in the mitochondria. That process is known as oxidative phosphorylation. When we consider that 2 moles of glyceraldehyde are generated from 1 mole of glucose, we can conclude that from the 1 mole of glucose, we generate 2 pyruvates, 2 NADH, and 4 ATPs. But don't forget, we already consumed 2 moles of ATP in the energy investment phase. We will talk more about this later. The energy investment phase consumes 2 moles of ATP per glucose. The energy generation phase generates 4 moles of ATP, 2 moles of NADH, and 2 moles of pyruvate. In anaerobic glycolysis, the NADH is used to reduce pyruvate to lactate through the action of the enzyme lactate dehydrogenase. This reaction is crucial so that NAD+ can be regenerated so that glycolysis can continue. Thus, in anaerobic glycolysis, the net ATP generation is 2. That is, 4 moles generated minus 2 moles consumed equal 2 net moles. However, under aerobic conditions, the NADH generated passes the hydrogen to the electron transport chain in mitochondria through either the malate aspartate shunt or the glycerophosphate shunt. The malate aspartate shunt is the major shunt in the liver. It is estimated that 2.5 moles of ATP are generated per NADH oxidase. Therefore, from the 2 NADH oxidation, we get 5 ATPs. If we add this to the 2 net ATP we get through substrate-level phosphorylation, it will add up to 7. Thus, from 1 mole of glucose, we get 7 moles of ATP and 2 moles of pyruvate under aerobic conditions. The pyruvates can then enter the mitochondria for the Krebs cycle reactions. You can watch my video on the Krebs cycle to gain more understanding of the cycle. Alternative fates of pyruvates. Different things can happen to the pyruvate generated from glycolysis, under different conditions. Here, we will look at 4 major things that could happen. 1. Under anaerobic condition, the pyruvate is reduced to lactate by lactate dehydrogenase. This reaction is very essential to regenerate NAD+, which is a substrate for glyceraldehyde 3-phosphate dehydrogenase, thus enabling the continuity of glycolysis. 2. Under aerobic conditions, pyruvates goes to the mitochondrial and is used to generate acetyl-CoA by the enzyme pyruvate dehydrogenase. Acetyl can then enter into the Krebs cycle. 3. During fasting, starvation or any other condition that could cause a lowering of blood glucose level, the process of gluconeogenesis begins in the liver and kidney. One of the reaction steps is the carboxylation of pyruvate to form oxaloacetate. The enzyme that catalyzes this reaction is known as pyruvate carboxylase, and it requires biotin as a cofactor. 4. Pyruvate can be transaminated by alanine aminotransferase, also known as alanine transaminase, to form alanine. It is a reversible reaction. This reaction can occur in the liver, but also occurs in the skeletal muscle during starvation period when muscle proteins are broken down and the amino acids undergo transamination reactions. Anabolic pathways associated with hepatic glycolysis. In the liver, glycolysis is connected to several anabolic processes. These processes utilize glucose to form other important products in the body. One of them is glycogen. In the fed state, insulin enhances glycogenesis, especially in the liver and skeletal muscle. Here, glucose 6-phosphate is converted to glucose 1-phosphate, which is needed for glycogen production. Enzymes such as glycogen synthase and branching enzyme are activated. You can watch my video on glycogen metabolism and glycogen storage diseases for more information on that. Secondly, the acetyl-CoA obtained from pyruvate is used to form fatty acids and cholesterol in the fed state. Likewise, the sum of the dihydroxyacetone phosphate generated in glycolysis is reduced to glycerol 3-phosphate, and this is used to form the backbone of triglycerides and glycerophospholipids. Addition of fatty acids to the glycerol 3-phosphate would eventually lead to the formation of these lipids. Thus, glucose can be used lipids in the fed state. Another important thing to know is that the glucose 6-phosphate produced from glucose can be diverted into the pentose phosphate pathway to generate NADPH and the pentose phosphates. NADPH has several important uses. One of them is reductive biosynthesis, where it is used to hydrogenate or saturate some unsaturated bonds during fatty acid and cholesterol biosynthesis. Thus, hepatic glycolysis is associated with anabolic processes in the liver. Regulation of hepatic glycolysis. Regulation of biochemical pathways, very crucial aspects of biochemistry for students to take note of. In the fed state, insulin stimulates processes that utilize glucose. Hence, insulin stimulates glycolysis, glycogenesis, fatty acid synthesis, cholesterol synthesis, lipogenesis and the pentose phosphate pathway. Insulin also inactivate all processes that produce or preserve glucose. Thus, insulin inhibits glycogenolysis, lipolysis, fatty acid oxidation, and ketogenesis. Epinephrine, glucagon and cortisol are known as counter-regulatory hormones. They oppose most of the actions of insulin. In the fasting state, glucagon and epinephrine inhibit hepatic glycolysis, glycogenesis, and fatty acid, cholesterol and lipid synthesis. They stimulate gluconeogenesis, glycogenolysis, fatty acid oxidation and ketogenesis. Thus, these hormones stimulate processes that preserve or produce glucose. Regulatory enzymes of glycolysis. There are 3 irreversible steps in glycolysis. These are the regulatory steps, and we are going to look at them one after the other. The enzymes that catalyze these reactions are displayed on the screen. 1. Glucokinase. Glucokinase has a large km for glucose. This is about 10 millimolar. Compare with hexokinase that has a low km of about 0.1 millimolar for glucose. Large km denotes little affinity. Thus, during fasting or starvation, glucokinase does not do catalysis. That is one of the reasons why the liver does not consume glucose during fasting. However, due to the large Vmax, which is high catalytic efficiency, of glucokinase, it can rapidly convert glucose to glucose 6-phosphate during the fed state. The glucose 6-phosphate can continue with glycolysis, or it can enter the pentose phosphate pathway or the glycogen synthesis pathway. In the fed state, insulin stimulates the production of glucokinase through transcription and translation, thereby enhancing glycolysis. In the fed state, liver takes up much glucose from blood and rapidly utilizes it for the aforementioned processes, thereby preventing hyperglycemia. Glucokinase is inhibited by fructose 6-phosphate. Note: defect in the glucokinase enzyme in the liver and/or beta cells of pancreas can lead to MODY, maturity-onset diabetes of the young. 2. PHOSPHOFRUCTOKINASE1. PFK1. PFK1 catalyzes the second irreversible step in glycolysis. The most important activator of this enzyme in the live is fructose 2,6-bisphosphate, which is produced by the action of PFK2 enzyme activity. Other important activators of PFK1 are AMP and ADP. Important inhibitors of PFK1 are citrate and ATP. Insulin, through a protein phosphatase, enhances the action of PFK2, thereby leading to the production of fructose 2,6-bisphosphate and activation of glycolysis. Glucagon and epinephrine, through the action of protein kinase A, activates fructose 2,6-bisphosphatase, which breaks down fructose 2,6-bisphosphate to form fructose 6-phosphate, thereby inhibiting glycolysis. Increased amounts of citrate and ATP is an indication of abundance of nutrient and energy. Thus, glycolysis is inhibited. 3. Pyruvate kinase. This enzyme catalyzes the third irreversible step in glycolysis. Pyruvate kinase is activated by fructose 1,6-bisphosphate. This is an example of feed-forward activation. Pyruvate kinase is inhibited by phosphorylation. Epinephrine and glucagon, through their hormonal actions, stimulate protein kinase A, pKa, which phosphorylate pyruvate kinase and makes it inactive. Insulin, on the other hand, stimulates a protein phosphatase which dephosphorylates pyruvate kinase and renders it active. Alanine inhibits pyruvate kinase allosterically. Now, let's look at the disorders of glycolysis. If anything tampers with glycolysis in the body, the mature red blood cells are among the first cells to feel the repercussion. This is because they don't have mitochondria and get their energy supply through anaerobic glycolysis only. You may wish to watch my video on glycolysis to know more about the importance of this pathway in the various tissues of the body. In today's video, our major focus is on hepatic glycolysis. Pyruvate kinase deficiency is the most common glycolytic pathway disorder. It accounts for about 95% of the glycolytic enzyme disorders, and it is autosomal recessive. Patients suffer from hemolytic anemia. Phosphorhexose isomerase deficiency takes about 4% of the glycolytic enzyme deficiency disorders, while the rest of the glycolytic enzymes take about 1% altogether. Arsenic poison can affect glycolysis in this way. Arsenic competes with inorganic phosphate as a substrate for glyceraldehyde 3-phosphate dehydrogenase, forming a complex that spontaneously hydrolyzes to form 3-phosphoglycerate. By bypassing the synthesis and dephosphorylation of 1,3-BPG, the cell is deprived of energy usually obtained from the glycolytic pathway. And that is it for today. Today, we discussed hepatic glycolysis. We touched the metabolic importance of the pathway, the anabolic pathways associated with hepatic glycolysis, the hormonal regulation of the glycolytic pathway, the regulatory enzymes of the pathway, the activators and the inhibitors. We talked about the energy yields of the glycolytic pathway and the alternative fates of pyruvate. We also talked about the associated disorders of the pathway, including pyruvate kinase deficiency and arsenic poisoning. I hope you found